started off his career at Vanderbilt University.
We can't hear you.
You can't hear me.
No, because the door keeps opening.
Oh. I guess we should show up on time. Let's see. We're just as bad as the students. Okay. Anyway, I'll speak on more loudly. So he got his start under David McCauley at Vanderbilt. His honors thesis on investigations of gene flow using molecular markers in white campion. He then moved on to Indiana University working with Ellen Ketterson. His dissertation was phenotypic integration of sexually selected traits in a songbird. After that, he moved on and worked with Butch Brody as a postdoc at the University of Virginia, so he's got quite the lineage to back up a lot of his work. Along the way, he picked up a few awards, the Office Phillips Award from the American Society of Naturalists, as well as the Jansky Award from the Evolutionary Society. Also, I think you had some NSF DBIGs and DRFs and things along the way, and he's currently funded to talk about this work here today, which I'm excited to hear about. So I'll let you go ahead and get started.
That's fine. It's been great being here today. I've had a great time talking to everybody, and I'm excited to talk to you a bit about what's going on in our lab right now. So as evolutionary biologists, one of the main things that we're interested in is explaining the origins of complex novel adaptations. So things like flight in birds, vertebrate vision, the vertebrate eye, venom delivery in snakes, and adaptation to extreme environments like we see in this cactus right here. So when I say complex adaptations, I mean an adaptation that is built of a lot of parts that function together. So we're not just talking about one gene changing in response to natural selection. We're talking potentially about a bunch of genes put together. And so one of the things that has obsessed me over my career, even though the list of things Mike said sounds like they don't go together, the things that the common thread that runs through all that work is I'm interested in how the parts of an organism fit together and how natural selection shapes those relationships. So when we're talking about these complex adaptations, the things we want to understand as evolutionary biologists are the process behind that. That's usually natural selection. And what are the building blocks that natural selection uses? So what are the molecular genetic changes that actually happen and change in response to selection? And then what's the history of those adaptations? So how precisely have those building blocks come together? So a lot of these adaptations are so complex that it's almost impossible to study them empirically, and it's even hard to fathom conceptually how we might perceive them coming together. So instead of focusing on a particular adaptation, I'm going to start off with something a lot more simple. I'm going to start off talking about baby toys. So when we think about how adaptations are built, one possible way is that adaptations that are complex, consist of a lot of parts, could be like this puzzle here, right? So in this puzzle, our objective is to get all of these shapes into the right spot. And we might think of these shapes as being the environment. And if you've worked one of these puzzles, you know it doesn't matter which one you put in first, right? You could start with the pentagon. As long as you get it into the pentagon hole, you'll be fine. Or you could start with the octagon. It doesn't matter. As long as you get them all in the right place, you solve the puzzle. So we might call this environmental determinism. Here the environment sets up the basis for natural selection, and the building blocks fall into place. It doesn't matter what order they happen. Alternatively, adaptations could come together more like this set of stacking rings. So with the stacking rings, if you're older than 2, you know that you have to start off with the blue one first, or else you're not going to get to the right solution. So if you skip the blue one, you are in rough shape, and you'll end up with something that looks like this. However, if you start with the same set of materials, the same initial conditions, and go in a different order, then you can reach this final beautiful structure here, which we'd call our complex adaptation. So this process, when history matters, when the order that something happens matters, we will call historical contingency. That means that what comes later depends on what happened in the past. And so one of the questions I'll be focusing on here is, do complex adaptations look like a puzzle, or do they look like a set of stacking rings? And to answer this question, we need to have an adaptation that is not so complex that we can't handle it, but we also need to have something that is not so simple that it's boring. So in order to do that, I'm going to be studying something that I think hits the sweet spot of complexity, the sort of Goldilocks zone for complexity, and that is evolution of resistance to prey toxins in snakes. So this picture here, it may look like an ordinary snake having his lunch here. We have, I should turn the middle lights down. Like I said, I was going to, you can see the picture a little better. Is that better? Yeah. Okay, so here we've got one organism is now lunch. This snake is eating its lunch. And this looks like an ordinary meal, but in fact, this is one of the most perplexing interactions in nature. And that's because this species right here, who's become lunch, is perhaps the most toxic animal in the entire world. That's the rough-skinned newt, Tarica granulosa. The most poisonous newt in the world of this species. We could grind it up into a milkshake and pass it down this row here, and then none of you would survive. There would be enough toxin in that newt to kill you and 15 or so of your closest friends. However, this species over here, the common garter snake, Thamnophis sirtalis, seems to be able to eat most newts, most poisonous newts, without any ill effects. So this guy might be slowed down a little bit, but he's still going to go about his business, even though he's eaten perhaps the most toxic animal in the entire world. So this raises the question, how have snakes evolved this complex adaptation? How are they able to eat poison? So to answer this question, I'm going to build on a couple of decades of work by a couple of generations of Butch Brody's here. So Butch Brody, Part 3, 3.0, was my postdoc advisor. His dad, Butch Brody Jr., has been working on poisonous newts since the 60s, and together they worked on this interaction since the 90s. So back to the newt again. Here's the newt, the rough-skinned newt, doing its threat display, which is called the Unken reflex. So they do this when they're threatened and they're exposing this orange underbelly to say, hey, I'm poisonous, don't eat me. And most animals should pay attention to this, because most animals cannot handle this. And that's because newts are chock full of this neurotoxin called tetrodotoxin. It's a small molecule, but it's a weird molecule, and we actually have no idea how it's synthesized, and we don't even really know whether the newt makes it itself. So it's known to be made by some bacterial species, but we don't know whether the newt has a bacterial endosymbiont or whether it makes it itself. All we know is that this particular species of newt has a whole lot of it. And here's the way it works. So tetrodotoxin binds into voltage-gated sodium channels, which are the proteins in your body that carry current along excitable tissue. So carry current in muscles and nerves and in your heart. And voltage-gated sodium channels have TTX binding sites right here around the outer pore. And those are not just there to bind TTX. Those are there to select which ions go through the pore. So these are the sites of the channel that keep sodium going through and other things not going through. However, tetrodotoxin is an insidious little molecule because it will pop right into that pore and plug it up just like a cork in a bottle. And these regions that I'm showing here in 3D structures are these P-loop regions. So this is a 4-domain protein. Each domain has a loop that goes down into the pore, forms this outer pore structure where TTX bonds. So if we want to study how the snake is resistant to the toxin, the way that we do this is to inject toxin into the snake And then you have an old man with a cigar who chases this snake down this racetrack here, and it's important to have the cigar. It always has to be there. And you measure how much the speed of the snake is reduced by the tetrodotoxin. And when you do that across a bunch of different populations, what you find is that some snakes are really slowed down by the toxin. So what we have here is resistance measured in how much in reduction in crawl speed. So the further the curve is to the left, the more it's slowed down by the toxin. The further it is to the right, the more it's resistant to the toxin. And what you're looking for is this 50% mark. So how much toxin it takes to slow the snake down by 50%. And since this TTX goes here on the x-axis is on a log scale, what you have is some snakes are orders of magnitude more resistant to tetrodotoxin than others. And these lines here are from different populations. So if you look at the populations spread out geographically, what you see is this mosaic of resistance across the landscape, where you have some places where the snakes are super resistant, like here in the Bay Area in California, you find the most resistant snakes. And up here you have some that are pretty resistant. And then other places resistance is lower. So a couple of blue spots here. The purple is not on this graph, but if you move outside of this range, and this range is the overlap with the rough skin newt, what you see is very, very low resistance. So this species is spread across North America, and if you took an eastern garter snake, you'd find very, very low levels of resistance to the toxin. But out west, where they overlap with the newt, what you find is a general rough match between the resistance of the snakes and the toxicities of the newts in that area. And what this suggests is we have a coevolutionary arms race between the snake and the newt that's playing out both over space and time. So as the newt becomes more toxic, the snake becomes more resistant, which imposes selection on the newt to become more toxic. And this escalates. So you have some places where both the newt and the snake are super toxic and super resistant, and then other places where the interaction is not as intense like that either. So how does this happen on a molecular level? Again, thinking about the snake. So we're fortunate with voltage-gated sodium channels that we can look at only a few amino acids in the whole gene and understand how resistance evolves. And that's because we have this very specific binding site for the toxin that's made up out of these few amino acids right here. So I'm going to simplify this as much as possible. I'm showing the smallest window I can. The red amino acids here are the ones that are most important for TTX binding. And by and large, if you change any of these amino acids, you get a change in the affinity of tetrodotoxin for the channel 4. And you can show experimentally how amino acids change TTX binding. So you can express these things in frog eggs and measure current flow in the presence and absence of tetrodotoxin and show that if you change, say, this F here, which is phenylalanine, to C, which is cysteine, that you see a 1,000-fold change in resistance of the channel. So if you look across space here in the snakes, what you find is that more resistant populations, so the hotter colors here are more resistant populations, have more substitutions in their P-loop regions. And in particular, it's for this particular gene, it's P-loop 4. So what I'm showing here is the skeletal muscle voltage-gated sodium channel, which is called NAV1.4, and populations in Willow Creek, which is the most resistant population right here in California, has 4 different substitutions here. And this gives you about a 600-fold resistance to the toxin. And as you remove these substitutions, you get less and less resistant. However, this one right here is still a lot more resistant to the toxin than this non-resistant form, which is in Idaho, a place where they don't overlap with the nuke. So at first blush, this seems like not a complex adaptation. It seems like I'm showing you one gene here that is responsible for everything. One gene explains all this variation in resistance across populations, and I'm not telling a very interesting story about complex adaptations. But it turns out that this particular gene, NAV1.4, is part of a family of other voltage-gated sodium channels. So NAV1.4 is found in the skeletal muscle, and this, it might look like rigatoni, but this is what I'm using for skeletal muscles. There are other genes that are found in different tissues of the body. So for instance, 1.1, 1.2, and 1.3 are found in the brain. 1.5 is found in the heart. 1.6 is found in a number of places, but for our purposes, the most important place is on the nodes of Ranvier. That's these little spots along the axon of peripheral neurons, like the long axons like motor neurons. So they are responsible for carrying fast current in motor neurons. And then you have a few more that are found in smaller neurons, typically sensory neurons. So these are 7, 8, and 9. So if a snake is going to be able to eat toxin, you would expect it to need to have resistance at all of these genes, not just this one right here, not just the skeletal muscle. So a bit about the history of these genes. It turns out that these evolved quite a long time ago. So we can trace the origins of these genes back about 450 million years or so, back to 2 different genome duplications in the early history of vertebrates. So that's what gives us these 4 different sets of genes on 4 different chromosomes. So these are the human chromosome numbers here. And then along the chromosomes, we've had tandem duplication to give us a few extra copies. But this set of 9 here is what all amniotes have. So amniotes being reptiles, birds, and mammals. They have all of these. So if you're going to be a snake eating tetrodotoxin, potentially all of these are going to be exposed. They're all very similar molecularly. They all have that same pore, and they're all doing basically the same thing, just in different tissues. One complexity here to keep in mind is that the brain is actually protected by the blood-brain barrier. So unless you're injecting tetrodotoxin directly into your brain, you're probably not going to necessarily need to evolve toxin resistance in your brain. However, snakes, if they're going to be able to handle a meal of tetrodotoxin, probably need resistance in all 6 of these genes out here. So the question I'm going to be asking is, do we find resistance in all of these genes? One other complexity to keep in mind before I go forward is that 3 of these are resistant to tetrodotoxin in mammals. So if you were to eat a newt, you would die, but your heart wouldn't stop, at least immediately. And that's because NAV1.5 is really, really resistant to the toxin, and so are these 8 and 9. If you'll recall from the phylogeny on the previous slide, those 3 are clumped together. So initially it was thought that the resistance of these 3 dates way back to that original split. And I'll show you some evidence in a bit that may not be true. But keep in mind, these 3 are called the tetrodotoxin resistant channels because they're resistant in mammals. We didn't know before we started doing this work here whether or not they were actually resistant in snakes. So to answer this question, we took a couple of approaches. The first was to do a scan of the bacterial artificial chromosome library, or BAC library, that Scott Edwards had constructed for Thamnophis sirtalis. And we did this in Scott Edwards' lab at Harvard with the help of Dan Janes, who was there at the time. And basically you put the bacterial clones on a filter, you put radioactive probes on there, and this is one of the filters, or a picture of one of the filters, and those little blips here are the radioactivity of where our probes are hybridizing to the clones. So you pick the clones that are the right ones to sequence, and then you put it onto a sequencer. In this case, we used a 454 that John Chukalovchak ran the sequencing for us. And then you get back a bunch of sequencing. We did this because we did not yet have the genome of Thamnophis sirtalis, but now we do have the genome of Thamnophis sirtalis, so it's getting ready to be published. And Todd Casto and Mike Frender are taking the lead on this. So from this initial scan of the back library, we got the sequences of 6 of these sodium channels. We were missing the ones that are tetrodotoxin resistant in mammals, and fortunately we were able to pull those 3 out of the genome. And it turns out that we were very lucky to have done this step first, because the assembly of the Thamnophis genome was a mess for a number of reasons, and our sequence from the back library scans in some cases was better than what we got out of the genome. Pieces were missing in the genome that we were able to detect over here. Okay, the next 3 slides I'm going to show you look just like this, and these are the results of both of our back library scans and the genome scans. And what we have here are the 4 p-loop regions, and at the top we have 3 of the paralogs, or forms of voltage-gated sodium channels that we're looking at. So first we're going to look at the brain ones. So I have here the place where it's expressed, and then the name of the paralog. And I've got mouse, anolis, lizard, and Thamnophis here. And if there's a difference between this mouse sequence and the other sequences, I'll show the amino acid change. If not, I'll show a dash. I will always show the ones that are tetrodotoxin resistant, even if they are the same across all the species. So here we see a whole lot of nothing. So there's a couple of little differences up here in domain 1 that don't confer resistance, but by and large, what you see is that a brain sodium channel looks the same in mouse as it does in anoles as it does in snakes. So as we might have predicted from the blood-brain barrier, these things have not evolved resistance in garter snakes. But if we look at the periphery, we find a different story. So here we have the muscle channel that I've already talked about, where we have the resistance popping up via substitutions in domain 4. And we've got 2 peripheral nerves, 1.6 and 1.7. And what you see here is that both of these are predicted based on their amino acid sequence to be resistant to toxin. So in 1.6, we have a change in domain 4 that's exactly the same as one of the changes we see in domain 4 of 1.4. This valine here should double the channel's ability to handle toxin. Over here, this assembly of substitutions in 1.7 occurring in both domain 3 and domain 4 are going to give the channel about a 600 to 900 fold bump in resistance. So we've actually tested these now to show that they make a very, very resistant channel. When you look at anolis, you also see the evolution of resistance here. And these changes have been tested by others to show that they're resistant to toxin. So this suggests that this channel may have evolved resistance a couple of times. So this looks like a completely different mechanism when we look at anoles versus when we look at Thamnophis. So we have at least 3 resistant channels. And looking at the last 3, so these are the ones that are tetrodotoxin resistant in mammals. So they're resistant in mammals because of this one change here, in the same position across all 3, the C and the 2 S's. So in 2 cases, the heart and this peripheral nerve over here, Thamnophis has the same change as in mammals, suggesting that it may have dated back to the common ancestor of mammals and snakes. However, when we look at this one in the middle, 1.8, Thamnophis is missing this S. it still has an R in this position, which gives you a slight bit of resistance, but it doesn't have the big bump that this S here, the serine, would give you. However, looking at the other, we find that Thamnophis has evolved increased resistance in this channel via a threonine substitution right there, and Anolis again has this AE in that spot. So this is going to bump up the resistance of this channel in Thamnophis. So this is a lot of information. To sum it all up, we can put it back on this picture and we can put some numbers on how resistant these channels are. So we saw that the brain channels are not resistant at all. They're protected behind that barrier. And then everything else out in the periphery is resistant to tetrodotoxin. So remember, skeletal muscle channels vary across populations, between 200 and 600-fold resistance. And then all of the other channels in the nerves and in the heart are resistant to some level of tetrodotoxin. Some of them are resistant to a ton of tetrodotoxin. So this suggests that we are dealing here with a semi-complex adaptation. We have 6 genes that have evolved resistance at some point to tetrodotoxin. So now we want to be able to take these building blocks and ask how they come together. So what is the history of this complex adaptation? And to do that, I'm going to focus in initially on 3 of these. So we're going to focus in on skeletal muscle and 2 peripheral nerve channels. And we're going to piece together the history of them using the comparative method. So our first initial hypothesis is that maybe these genes have all evolved resistance in response to eating newts, in response to this ongoing coevolutionary arms race that snakes have with newts. So if that were true, what we would expect to see is that if we look at populations out here on the west coast that vary in their resistance and compare them to populations out here in the east that don't eat these really toxic newts, what we should see is a difference in these peripheral nerve sodium channels. So we just looked initially at 3 different populations. So one up here with medium levels of resistance, one here with high levels, and one out here with very low levels of organismal resistance. And what we found was the same gene sequence everywhere we looked. So these 2 channels are fixed in the species. All Thamnophis sertalis have resistant NAV16 and NAV17. So we knew that to piece together the history of this adaptation, we'd have to move the lens out a bit further. And so we just decided to move it out as far as we possibly could. So we sequenced 79 species of snakes, and Megan Cobiella, who is now a grad student at the University of Minnesota, was responsible for a lot of the bench work in this project. So over here we have all the species of snakes we sequenced, 79 of them. And we're aided in being able to do this by having 4 genomes of snakes that were provided to us by Todd Casto. So we had a blind snake, the Ramphotyphlops, a boa, a python, and a cobra. And we used these to design primers to pull out sequences in other snakes. And then we added to the tree, let me point out a couple of groups over here. So we focused most of our effort on this big group of snakes called the advanced snakes, or Colubroidea. And Thamnophis sertalis is down here in the bottom. So here's the big group of Thamnophis. And within Colubroidea, the main families we were focusing on were the vipers, Viperidae, Lapidae, that includes the cobra, and then Colubridae, which is the biggest family of snakes. There's a ton of snakes in it. Within Colubridae, there's a bunch of groups that probably are big enough to be families on their own. We have the Colubrians here, which include your rat snake and your king snake. The Dipsodines here, which include hognose snake and a few other things. And up here, the Natricines, which have our friends, the garter snakes and their relatives. So we added to this 2 sequences of lizards from published genomes and also, why does it keep doing that? There we go. One bird and one turtle as our outgroup. So I'm going to simplify this phylogeny here, we're going to prune it down to something manageable that we can actually look at. And what we'll do is trace the evolution of resistance in these three channels across time. So this phylogeny here is calibrated to time, and we've got millions of years before present. Up here, there's a dotted spot here because I didn't want to go all the way over here into this quote for that end of the phylogeny. So we got, truncated in that spot. And we'll build up the regions with TTX resistance right here in this box, starting with the earliest one. So it turns out that NAV 1.7, this is the channel that is found in small neurons, and we found to be very resistant in garter snakes, evolved its resistance a very, very long time ago. So this substitution right here, this N, this asparagine, which is found right here, gives the biggest bump in resistance to the channel. And this substitution occurred 170 million years ago or earlier. And if you'll notice here, this is outside of the snake clade. So snakes began right here. One of our lizards had it. The other one, Anolis, had lost it. It's this dotted line here. But almost every single snake that we looked at had that substitution. So almost every single snake is going to be resistant to tetrodotoxin at this channel. We had a couple of losses here in a couple of species, but most of them kept it. And these other two changes, this G and the Y that occur in domain 4, popped up quite early too. So they popped up in the common ancestor of this large group, the advanced snakes, the Colubroidea. So by 60 million years ago, we have a very, very, very resistant channel at NAV 1.7. And we also have this other change in domain 3, which, based on our data, appears to date way back into the Permian, 278 million years ago. So it's found in birds and turtles as well as in all of these snakes and lizards. The second channel to evolve resistance was NAV 1.6. So this is the one found at your nodes of Ranvier on your long myelinated axons. And this happened 4 different times, and it happened only in lineages that had the domain 4 channel substitutions. Whoops. I'm trying to go backward. It won't let me go backward. Try again. All right. Yes. It happened only in the lineages that already had these substitutions here. So we have now 4 origins of NAV 1.6, and this happened every single time via the same amino acid change. It's a valine in domain 1 or domain 4. So it happened once in the vipers, once in the elapids. Or twice in the elapids. Sorry. It's the cobra group here, and then once in the common ancestor of dipsodines and matrices. You'll see that it's lost a couple times within that group. So importantly, this includes Thamnophis. And then finally, within these groups of snakes that already had 2 resistant channels, we see 5 different origins of resistance at NAV 1.4, the skeletal muscle channel. So I'm highlighting the one that we've already explored here, Thamnophis sertalis. These other 4, I'll show you a little bit more about in a second, evolved resistance independently via different amino acid changes. Okay, so what we seem to have here is a stepwise buildup of resistance across paralogs in this gene family. So what could be driving this? So we know that newts evolved tetrodotoxin about 45 million years ago. All different species of newts have some tetrodotoxin. So it's tempting to suggest that all of this is driven by interactions with tetrodotoxin in the diet. However, these things happen way too early to possibly be driven by newts. And it's unlikely, even though there's tetrodotoxin in other amphibians, it's unlikely that it derived from eating amphibians in the past. It's likely driven by other sources of selection that I'll speculate on in a second. But these right here, the change in 1.6 and 1.4 may have been driven by newts in the diet or other amphibians in the diet. So one piece of evidence to support this is that 1.6 started evolving resistance shortly after the origin of newts, and all of these groups here overlap in space and time with ancient newts. Another piece of evidence is that these groups of snakes here include the species of snakes that are the most common eaters of amphibians. In particular, the groups down here, dipsodines and natricines, a lot of them are amphibian specialists. So this suggests that maybe tetrodotoxin or a similar toxin in the diet could have driven the evolution of 1.6. So as I said, 1.4 evolves 5 times within within these two groups, or within these 4 groups, dipsodines, natricines, elapids, and vipers, but actually all the origins are here within dipsodines and natricines, sorry. So only in the groups that have NAV 1.7 and NAV 1.6 resistant do we find these origins of NAV 1.4. All of these species here, as shown by work by Chris Feldman, that was published in 2012, all of these species are predators upon species with large amounts of tetrodotoxin and seem to be in ongoing coevolutionary arms races with their prey. And so you'll see up here that all of these are different independent origins of resistance in this channel. And we can show using some statistics that the origin of resistance in this channel is contingent on the earlier evolution of resistance in NAV 1.6. So we use this phylogenetic contingency test to show that it's significantly more likely to get resistance in 1.4, and hence significantly more likely to get these coevolutionary arms races when you already have the basal resistance from the other channels. So what drove resistance in this very early channel, NAV 1.7? Well, I told you that it's found in sensory neurons. One possibility is that it's modified for other purposes other than tetrodotoxin eating. So we know that when you change the pore, you often change the function of the channel. So you change how much current comes through or how selective it is for different ions. And one piece of evidence that suggests that it could have been modified in reptiles for other physiological reasons, and you get tetrodotoxin resistant as a side effect, is that this channel is found heavily expressed. In fact, it's the only channel found in the vomeronasal organ, or the Jacobson's organ, of snakes. And it's possible that this channel could have been tweaked for its role in olfaction. It's also important in olfaction in mammals, so it's retained that role. But snakes and a lot of their lizard kin do olfaction a little bit differently. And it's possible that you might want a slower channel or a slightly less selective channel to mediate the signals of olfaction to the brain when you're doing it with your tongue. However, that's just idle speculation. So when we put all these things together, we see a nice story of buildup of resistance in these channels over time. NAV 1.7 evolves resistance first, and this gives you a low level of organismal resistance. So it's not likely to help you survive if you eat something tetrodotoxic. but probably it's going to protect you against the sort of numbing sensation that you would get, the sensory aversion that you would have to eating something tetrodotoxic. And so it's going to predispose snakes to eventually become predators of tetrodotoxic animals. And it could be selected for by many reasons, but potentially its role in olfaction. The second channel, NAV 1.6, evolves resistance much later, and it's happening only in groups that are eating amphibians. This is going to give you a moderate level of organismal resistance to tetrodotoxin. So this means that once you have peripheral nerves that are resistant to tetrodotoxin, a small amount of toxin in the prey is not going to be effective against you as a predator. So it takes only a little bit of toxin to block a nerve. It's going to take a lot of toxin to block a muscle. So if snakes are coming in with resistant nerves, a tiny bit of toxin is not going to be an effective defense anymore. And so this is going to make some snakes good selective agents on their prey to evolve more toxin. So what this should do is occasionally set off coevolutionary arms races with prey, leading to the evolution of eventually resistant skeletal muscle and very high levels of resistance at the organismal level. And this will happen only in select species where you have an intimate relationship between the prey and the predator. So quickly, I want to mention what we found. We have some very early results looking at the history of NAV 1.5 and these other two channels that are very highly resistant in mammals. So we don't have the resolution that we have for the other channels. What we have is just a handful of genomes. But what we can say definitively is that the resistance in 1.5 and 1.9 goes way back. So it goes back to around 320 million years ago, the common ancestor of snakes and mammals. So most of the species in this tree have retained those two substitutions that give you a very high level of resistance in those channels. Why, we don't know, but they have. And NAV 1.8 actually wasn't very resistant at its origin. So most of the species here in the tree don't have a super resistant version of NAV 1.8. It's resistant to a little bit of tetrodotoxin, but it's not resistant to a lot. And we see an elaboration of its resistance in 4 different groups. The mammals, which appear to have maybe copied that S, the serine, from one paralog to another, and then via 3 different mechanisms and 3 different groups of reptiles. So I have a student who is plowing into this now to try to figure out the history of this one particular substitution that we see in garter snakes. We can't say much beyond that it evolved past the split with cobras, which is a long period of time. So you need a lot finer resolution to figure out when this happened. No! Okay, I'm getting a new pointer. All right, so we went into this thinking that maybe the evolution of resistance at the organismal level in snakes is more like a puzzle. So as long as you had all the pieces in place, you would be fine. And it's probably driven by the environment, driven by what you're eating. But what our results showed us is that this complex adaptation is more like one of these stacking toys. So you're not getting the high levels of resistance evolving that you need skeletal muscle channels to be resistant for until you have the base established. So low levels of resistance that are provided by peripheral nerves are going to eventually provide the conditions to lead to coevolutionary arms races. And this takes time. So what we see playing out contemporarily on the West Coast between newts and snakes builds upon 300 million years of evolution that have stacked the rings in the favor of such coevolutionary arms races. So this is all well and good. I've told a nice little story about how snakes have evolved resistance to toxin, but you might be thinking that this is, what he has there is just an N of 1, right? He showed that this gene family evolves in a certain way in one particular lineage. So, big deal. What does this tell us? So, what we would really ideally like to be able to do is now take this scenario of stepwise buildup of resistance and apply it to another group. And so, this is what I'm doing in my current project. We are asking whether we see the same pattern, whether we have parallel evolution of this historically contingent pattern in other lineages that should be selected in similar ways. And to get at this question, we're using both lizards, which although they don't have a ton of amphibian eaters, there are a few, including this monitor lizard here, and in birds. So there are more species of birds to work with, and also more species of birds that eat amphibians. In lizards, we have an extra bonus in that we can use the evolution of the gene family in lizards to further reconstruct the early history of what's happening in snakes. But these provide us a testing ground for our scenario that we built up in snakes. So I just want to show you some early results that we have. These are some very preliminary data from lizards. What we did is just started to sequence NAV 1.7 in a lot of lizard species. And before this chunk right here, so we had Ophosaurus and we had Anolis before, and then we had snakes before. We filled in a bit of the tree here, and we've gotten some that are outside of that group. So we have a couple of geckos up here, and we've got a couple of legless lizards here. And what we see here is that the origin of this N, we can push back maybe a couple 12 million years more to about 182 million years based on this particular reconstruction, because it shows up here as well. But also, this shows us that this particular spot on that gene is evolving a lot. And we appear to have resistance blinking on and off a number of times within lizards. So this gives us some variation to work with, and potentially might set up a comparative method study where we try to get at what really drove the evolution of resistance in this channel in the first place. In birds, we're focusing in primarily on herons and kingfishers, and also to a lesser extent birds of prey. And herons and kingfishers actually do eat newts. In particular, the great blue heron will eat the rough-skinned newt, and this guy has caught a rough-skinned newt here, and the poor guy is doing his Unken reflex, and it's not going to do him any good. He's going to get swallowed by the heron. With the birds, one advantage that we have is that there's this huge avian phylogenomics project. So this gives us a bunch of genomes to work with, and this is what we have so far. So my postdoc, John Abramian, is working on this now, and this is a ton of information here. And I'm not going to show you everything, but what this is are all the genes, all 9 genes, from around 60 different genomes. And black here means non-resistant, white means we couldn't find that particular sequence. Each of these is the domain of that gene. And then the color shows us that resistance exists in that channel. So the big pattern to notice here is that you have linking on and off of resistance in these peripheral nerves So some of the species of birds seem to have those basal level resistance things in place, just like snakes, but you never have resistance evolving in 1.4 and 1.6. So this suggests that these two really are more constrained and are going to need selection from some outside force to evolve resistance. We got a couple of cases of resistance even popping up in the brain channels. From here, we're going to take these genomes and do targeted sequence capture using baits that we designed from all of these genomes, and hopefully pull out sequences from a lot more birds, particularly focusing in on those herons and kingfishers, which are really eating a lot of amphibians. So I'll close here with a plea. We have funding, and we're looking for people. John just got a job. So this is what happens when you hire good postdocs. They get jobs and leave. So I'm looking for a postdoc to replace John, continue this work. I'm also looking for a graduate student to work on this project. So if you are one of the people that would like to work on this project, let me know. Or if you know a person who would like to work on this project, let me know. And with that, I want to thank all the people who have helped out along the way. Many of these are co-authors that I've mentioned throughout the talk. I want to thank NSF for funding, and thank you all for coming to my talk and listening. I'd be glad to take some questions. Yeah.
In the skeletal muscle mutations, do you see any change in the normal function of the sodium channels?
Yes. So I'm going to give you a general statement. In general, these things kind of mess up the channel. So it interferes with the general function of the channel in a number of ways. So including reducing conductance, reducing selectivity. So I can't off the top of my head spit out what each of those things do, but I can say that the most resistant populations of snakes have skeletal muscles that are slower twitch, and they're slower overall. So baseline speeds in the most resistant populations are slower. They get knocked down a lot less when you inject tetrodotoxin, but they start out slower. So this suggests that having those substitutions there does impose some cost on. Yep.
I just wanted to say I really like this story. I can't wait to see what comes out in the future. I had read a paper a while back ago called Puff Puff Pass,
Yeah.
and it was about dolphins taking tetrodotoxin from fugu from the pufferfish, and they were sort of recreationally using it to make people think they get high. So I was just wondering if you look at this, I mean, whether it's true or not.
It's got to be true.
So, I was wondering if you've looked to see if dolphins, for example, have resistance in any of those specific channels, and B, if there's any place where you could think of where sensitivity, sensitizing the channel may have greater benefits than generating resistance.
That's so that is awesome. I'm going to look that up. I have not looked at dolphin sodium channels yet, but I will. I think is there a dolphin genome? I think there is. There's tons of mammal genomes, so I think there might be a dolphin. So it's interesting to go take a look. So I know that I've heard stories of people like licking newts for fun, right? Just Easter newts. And the newts around here, the Notophthalmus, have lower toxin levels. So you're not going to, if you lick a newt around here, you're probably not going to die. You probably don't want to eat it, but you'd probably be okay. So I would not be surprised if that were true about dolphins. There is, so to answer the second part of your question, there's a related story in grasshopper mice that eat scorpions. And what has happened there is that their sodium channels have evolved in such a way that the scorpion toxin, which normally it works in a different way. What it does is holds channels open. So it holds channels open in pain neurons and typically affects one particular channel and causes this intense pain sensation. And what has happened to their channels is that a different channel, that one still works. It still opens the channel. It still initiates the pain sensation. But a channel downstream on the neuron now gets blocked by that same toxin. So they get the initiation of the pain sensation, but it gets dulled by the toxin blocking a channel downstream. So I initially thought that, so one of these, I'm not going to go back to this slide, but one of these channels, NAV 1.8, that's the one that carries that pain sensation. And I initially thought, well, maybe garter snakes don't need to evolve resistance in that. It will just be like when it gets blocked, when they eat a newt, it's just like, taking an Advil or something, or taking morphine. It's just going to block the pain sensation. Turns out they did evolve resistance in that channel. But I think that, at least for snakes, they keep it in their system for so long that probably it's going to screw up the function if it blocks it. That was a long answer to your question. Do we have time? Yeah.
So is there a fitness cost to having the resistance? Because why don't all the different populations of garter snakes in California have the same level of resistance?
Yeah, I think there is a fitness cost. So it does slow them down. So you, at least
So
the
it's like
skeletal
a
muscle
predator
one does
escape or...
Yeah. So I would suspect that if you took the newts away completely, and there's still genetic variation across all these populations, if you took the newts away, selection should flip, and it should favor the non-resistant channels. It'd be nice to do that experiment.
Yeah, do they have fewer mutations, less resistant populations in the same resistance channels?
Yeah, so for that skeletal muscle, the more resistant one has like 4 substitutions there, the less resistant has 2, and then the non-resistant has 0. But there's still, even in the very most resistant population, there's still segregating variation, and so you find non-resistant alleles in that in the most resistant population.
So have they reverse mutated? Is there a population phylogeny?
There is, and
Is it
one of
fine
Butch's
enough?
students is working on this, and I don't know all the details, but I think he's found that some of these things have popped up more than once from the population phylogeny. So there is just a mitochondrial phylogeny of the populations, which they used to make that tree that I showed, but they now have a gene tree of all the different alleles out there, and I think they've showed now that there's some multiple origins of things. But that's about all I know. Yeah.
In the resistant snakes, how do they purge the combustion? Do they metabolize it, or how do they excrete it?
It just eventually comes out, and so they eventually excrete it. It stays in their system a long time. So it stays in their system, I believe, so there's a study that looked at this, and the number that sticks in my head is 45 days on average. So it stays there a long time, which you might expect gives you an advantage if you have predators around that might eat you. And, so the hypothesis is that they are actually using that as a predator defense of their own. And so in support of that, there are some of the populations that are the most resistant are also more brightly colored. So garter snakes vary a lot in the coloration, and the red garter snakes, like I showed in the first slide of my talk, are found out west, and they're very highly resistant populations. So it suggests maybe they're aposematic, they're advertising to their own predators. Thanks very much.